Hi, my name is Daniel Berenberg. I'm a research analyst at the Flatiron Institute in the Bonneau Lab, and today I'm going to be talking about our work in learning sequence structure and network features for protein function prediction and how to integrate those features. Presently, we have access to a large swath of protein data. The PFAM database is composed of over 10 million sequences. We have over 165,000 experimentally determined structures. We have close to 400,000 high-quality homology models. And the string network database contains billions of interactions across millions of proteins for thousands of organisms. We expect that these different data modalities indicate different aspects of protein function in a complementary way. Namely, the protein sequence ought to contain conserved motifs that are likely functional. The positioning of secondary structural elements in 3D space provides some kind of functional specificity. And finally, the system-level context offered by protein interaction networks illuminates higher-order information, such as biological pathways, functional partners, etc. The problem is that these data are largely unannotated, and so combining them in a supervised learning context is likely fruitless. So what we propose is to use unsupervised learning. Namely, we train 3 different feature extractors on the 3 different source modalities and use the extracted features in order to train a downstream function prediction model. So I won't go too much into the details of the underlying models, but at a high level, the structural features are obtained through a graph autoencoder, which takes as input the thresholded distance matrix or contact map of the protein and is tasked with reconstructing it. The sequence features are obtained through a language model, which is tasked with predicting the next residue given the previous sequence portion. And the network features are trained by training a multimodal autoencoder, which takes as input multiple interaction graphs and is tasked with reconstructing them. But it's not about the different inputs and outputs of the models, but rather the internal representation that is learned during training. We can examine these representations by viewing a dimensionality reduction of the constituent feature spaces. And so we can see that the sequence features on the bottom left of this slide, they're clearly clustering DNA binding proteins fairly well and some transferase activity. The network features are also clustering those 2 terms fairly well. But unfortunately, the structural features are basically just a large mass. And although there is some coagulation of these proteins, the space is definitely not separable. And so clearly there's something going on with the structural features. We can kind of confirm this by training up multimodal integrative models on varying subsets of the modalities. And here we can see that, in fact, in orange, the multimodal sequence network and structure model performs worse than the network-only model. So clearly the network features are only adding noise or redundant information there. We can further investigate this phenomenon by comparing sequence and structural features. Here at the top, we can see that the functions for which structural features perform better than sequence only perform better to a very marginal degree, and in fact could be even statistically insignificant. Additionally, we know through previous lab work that the sequence features of this same language model are capable of reconstructing multiple different types of contact maps. And so what this means to us is that sequence features do contain structural information, so these feature sets are redundant. And clearly, the benefit of sequence features or structural features definitely is outweighed by its detriment. So moving forward, we can just eliminate structural features for the time being and continue to use sequence and network because there is going to be no information loss in terms of the modalities. We integrate sequence and structure by using another recent lab method called the Multispecies Maxout method, which leverages the power of network features and addresses the data availability crisis of network methods by combining interaction networks across multiple species. This is done by first computing blast similarity or blast similarities across the separate nodes of each network, and using those similarities as input to the isorank algorithm, which will build rich network features based on the topologies of the constituent networks. So this method is already an integrative method, clearly, because it uses blast similarity, but in order to further extend its integrativeness, we concatenate the sequence features with the isorank features in order to perform the protein function prediction task. And here we can see that such a concatenation does result in an increase in performance. On the top figure in both biological process and cellular component, we can see that the language model isorank concatenation method in orange outperforms the isorank only method in violet. And in the molecular function ontology, we can see that there's at least no degradation in performance, which gives us an indication that these features do not conflict and they do not assert any biases like we saw in this structural features where there's actual degradation in performance. That claim is further supported below, where we see that the contribution of sequence features to the sequence and isorank model largely includes the contribution of network features. So in conclusion, sequence structure and network features do characterize protein function to varying degrees. The network features are by far the most informative, and so leveraging them is important. The unweighted binary graphical representation of the protein structure was not the most informative and did not characterize the functional specificity well. But the basic integrative approaches between sequence and network did show some slight increase in performance. So with that, I'd like to thank my collaborators at the Flatiron Institute and at NYU, and thank you all for listening.